So, hello, my name is Ruul Delwel. I'm from Rotterdam, from the Department of Hematology of the Erasmus University Medical Center. Today I'm going to give you some tips on different talks and sessions that were dedicated to the epigenetics of normal and malignant hematopoiesis. The first session that I want to highlight is one that I chaired myself, which was on epigenetics. And there were two speakers. One was Alex Rajewskaya, and the other one was Francesco Boccolata. And Alex really explained very well, what the major chromatin modifications are that most people study nowadays, and how you can learn from that. How leukemia cells may be derived at chromatin and how important the chromatin level modifications are. And in fact, she really focused on a very specific small subtype, but with that subtype, she really explained very, very well, how changes in modulators of chromatin can lead to leukemia. Francesco explained very nicely the importance of the folding of the chromatin and how that can affect alterations in gene expression, and how it also helps to understand, in this case, different subtypes of leukemia, T-cell leukemia. Iñaki Martin Subaru, he is now you can almost say sort of a guru of methylation in leukemia. He really studied methylation leukemia already for many years, and he very nicely explained that first of all how important methylation is for the biology of disease, because by methylation of DNA you can turn off genes, and by demethylating you can turn them on. But he also shows very nicely how the methylation can now also be applied for the diagnostic and the prognostics of those diseases. Furthermore, there were two sessions on nanopore sequencing, and also there methylation was very important. And nanopore sequencing is a very interesting technology, which is still further in development, but it's a way of sequencing where you can really read very long sequences of DNA. And the interesting thing of nanopore sequencing is that with those long read sequences, you can check, you can detect mutations, deletions, alterations, any alterations, but you can also look in that same read, can determine whether regions are methylated or not methylated. And that technology will probably be applied more and more. And there was one very nice talk where in one of those sessions, where they really showed by applying this technology that a unique new subtype was identified that was still unknown, of which the biology was still unknown. So go to those sessions, and the two sessions are EHA2026 and EHA2026. So please go to those two sessions. EHA2026, which was called nanopore-based genomic and epigenomic profiling, and the other one was nano-leukemia, both about nanopore sequencing and how that can be integrated in current, nowadays, molecular analysis. So with that, I wish you a lot of fun listening to those talks, and wish you a nice day.